In this part 2 of Neoplasia lecture, we will look at benign and malignant neoplasia. We will concentrate on the characteristics of benign and malignant neoplasms, such as differentiation and anaplasia, nuclear features, rate of growth, local invasion, metastasis, telomerase activity, and monoclonality. First, we look at differentiation and anaplasia. Differentiation is defined as the extent to which the neoplastic cells resemble their normal cells morphologically and functionally. Generally, a benign neoplasm are well differentiated, whereas a malignant neoplasm can be well differentiated, moderately differentiated, or poorly differentiated. They can also be undifferentiated. Anaplasia is defined as the lack of differentiation. Now we look at the concept of well-differentiated neoplasm. First, we look at the normal adipocytes. Adipocytes are large globular cells distended by lipid content, and in between them, they have rich network of capillaries. Okay. Now we look at the dipoma, which is a benign neoplasm of adipose cells. We mentioned generally benign neoplasms are well differentiated, which means that the neoplastic cells resemble their normal cells morphologically and functionally. Under the microscope, the lipoma cells will look very similar to the normal adipocytes. The photograph on the left shows normal colonic mucosa. The crypts, although somewhat tangentially sectioned, are relatively uniform, parallel, and evenly spaced. The photograph on the right shows adenocarcinoma of the colon. The crypts are irregular in outline, they branch and are not evenly spaced. The cells lining the crypts appear dark due to nuclear hyperchromasia combined with a marked increase in the nuclear cytoplasmic ratio. The neoplasm still forms identifiable glandular structure with the presence of the lumen, similar to what we see in the normal colonic mucosa. And this is what we call a well-differentiated adenocarcinoma. Look at this normal squamous epithelium. It is composed of epithelium that produces keratin, that has intercellular bridges, and sometimes you can see keratin pearls. The next picture shows the neoplasm, which is a well-differentiated squamous cell carcinoma. You can still see the squamous epithelium features with identifiable keratin production, such as at the arrow, keratin pearls, and intercellular bridges. And that is why we call this a well-differentiated squamous cell carcinoma. It still has the features of squamous epithelium, even though it is a malignant tumor. Now we look at the concept of poorly differentiated tumors. Okay, and we usually see this in a malignancy. Remember, a malignant tumor can have a range of differentiation from well differentiated, moderately differentiated, and poorly differentiated. Look at the normal squamous epithelium and the normal glandular epithelium. And now we look at the neoplasm, the poorly differentiated carcinoma. It doesn't have any identifiable structure that points towards glandular or squamous differentiation. The only way we can know that this is a carcinoma is when we do an immunohistochemistry using antibodies that can detect the presence of epithelial antigen. Okay, so this is a poorly differentiated carcinoma because there is no identifiable structure that points towards any differentiation. Now we look at the concept of anaplasia, which is lack of differentiation. The cells in this picture shows marked pleomorphism, which is variable shapes and sizes of the cells and their nuclei. So the cells are pleomorphic, and it doesn't show any differentiation towards any identifiable structure. Yeah? So we don't see any squamous differentiation or whether there is any glandular differentiation. So this is anaplasia, lack of differentiation. Remember, we said differentiation is whether the cells, the neoplastic cells, resemble the normal tissue, either morphologically, how it looks like, or functionally. Okay, so benign neoplasm have normal function. For example, adrenal cortical adenoma still produces adrenal cortical hormones when it is neoplastic. Malignant neoplasm have variable function according to the degree of differentiation. Next, we look at nuclear features. Benign neoplasm usually have nuclear to cytoplasmic ratio or NC ratio that is close to normal. That means there's a lot of cytoplasm and there is small nucleus. The mitosis have normal mitotic spindles, whereas malignant neoplasm has nuclear to cytoplasmic ratio that is increased, and the nucleoli are usually prominent. Mitosis may have normal or atypical mitotic spindles. Look at the normal cell. There is abundant cytoplasm with small nucleus that is sitting nicely at the basement membrane. Compare that to the malignant cell with increased nuclear cytoplasmic ratio. You can see that the nucleus is large in comparison with the cytoplasm. And you can also see that the nucleus are crowded in the malignant cells. Compare that to the normal cell. Look at the typical mitosis. It can show metaphase, anaphase, or telophase. And from one cell, it divides into two cells. So in the typical mitosis here, you can see the anaphase and the metaphase here. Whereas here, you can see atypical mitosis, which has tripolar shape, such as the Mercedes-Benz logo. So one cell is trying to divide into three cells. Now we look at the rate of growth of neoplasms. Benign neoplasm usually is slow growing, whereas malignant neoplasm have variable growth rate. They are usually fast growing. How long does it take to produce a clinically detectable neoplastic mass? It is estimated that transformed cell must undergo at least 30 population doublings to produce 10 to the power of 9 cells, which is the smallest clinically detectable mass. Benign neoplasm do not invade. They grow and expand slowly with a rim of fibrous capsule that separates them from the host tissue. So this is an adrenal gland. And you can see that in the center, it has a tumor. It is very round, also described as well circumscribed. They do not invade into the surrounding tissue. Okay? The capsule is derived largely from the extracellular matrix of the native tissue due to atrophy of the normal parenchymal cells. Compare that to a malignant tumor, which is usually having some local invasion. So the benign tumor just now, they grow in an expansile manner, but a malignant tumor will invade into the surrounding tissue. Okay, so there will not be any capsule separating them from the surrounding tissue. So invasion is the second most important criteria for malignancy. So this is the invasive lung carcinoma. So the tumor margin is irregular and not circumscribed compared to the benign neoplasia we just seen just now. For invasion to occur, a sequence of events has to develop. First of all, there should be loss of intercellular adherence with the non-production of E-cadherin. Then the cell will attach to the laminin, which is the glycoprotein in the basement membrane. The cell will then release type 4 collagenase, which will digest the basement membrane, which is made up of type 4 collagen. The cell receptors will attach to fibronectin in the extracellular matrix, and the cell will produce cytokines that will stimulate locomotion and protease to dissolve the connective tissue. And the cells will produce factors that stimulate angiogenesis for the supply of its blood to the malignant cells. Okay. Metastasis is the spread of cancer from one part of the body to another part of the body. Benign neoplasm do not metastasize, and malignant neoplasm may metastasize to other parts of the body. There are three pathways of metastasis: seeding in body cavity, lymphatic spread to lymph nodes, and hematogenous spread via the blood vessels. This is an important terminology in terms of metastasis. Primary site of malignancy is where the origin of the malignant cells. Secondaries of metastatic tumor are the site of metastasis, away from the primary site of malignancy. Metastasis is often more common than primary cancer in lymph nodes, the lungs, liver, and bone. Note that all these organs are richly vascularized organs. They receive a lot of blood supply from the body. Lymphatic spread is the usual mechanism of spread for carcinomas, which are the malignancy of the epithelium. Regional lymph nodes are the first line of defense against the spread of carcinoma. If the nodal architecture is destroyed, the malignant cells enter the efferent lymphatics, which drain into the bloodstream, and from the bloodstream, the malignant cells can metastasize to distant organs such as lung, liver, bone, and other organs. So you can see that these are the normal looking lymph node, with the lymphoid tissue and the germinal centers, and you can see this is a different type of tissue that is not commonly seen in the lymph nodes. This is foreign tissue from the lymph node, and this is the metastasis. The second pathway of metastasis is via the hematogenous spread. This is the usual mechanism of spread for sarcomas, and the cells enter the veins and metastasize to the organs. The cells can enter the vena cava to metastasize to the lung, or portal vein to metastasize to the liver. Some malignancies have both lymphatics and hematogenous spread, such as renal cell carcinoma and hepatocellular carcinoma. The third pathway of metastasis is seeding into the body cavity. We have three main body cavities, which are the peritoneum, pleural cavity, and the pericardial cavity. So seeding into the body cavity is where the malignant cells exfoliate from a surface and implant and invade the tissue in the body cavity. For example, primary surface-derived ovarian cancers can commonly exfoliate from the surface of the ovary into the peritoneum cavity, and they seed on the omentum, which is present in the peritoneum. Peripherally located lung cancers commonly seed in the pleural cavity, and glioblastoma multiforme commonly seeds in the cerebrospinal fluid, causing spread to the brain and the spinal cord. Once malignant cells invade into the normal tissue, they can penetrate to the small lymphatics or the vascular channels. Then they can release the neoplastic cells into the circulation. The cells has to survive in the circulation, and they usually stop in the capillary beds of the distant organs. They embolize to the distant organs capillaries, and then they penetrate the lymphatic or blood vessel walls, and this is followed by growth of the tumor cells. Yeah, so that is how a tumor migrate, metastasize from the primary tumor into the metastatic tumor. Yeah, the primary tumor is in the primary organ, and then the tumor spreads into another organ. Now we look at the telomerase activity. First, what is a telomere? A telomere is a compound structure at the end of a chromosome, just like in the pictures. It gets shorter with cell division. The cell can normally divide only about 50 to 70 times, with telomeres getting progressively shorter. It is the cell's internal clock to count the number of cell divisions that has been made and stops further divisions after a set number. The enzyme telomerase adds bases to the ends of telomeres. In young cells, the telomerase keeps telomeres from wearing down too much. As a cell becomes cancerous, it divides more often and its telomeres becomes very short. If it becomes too short, the cell may die. Cancer cell can activate telomerase, which prevents the telomeres from getting even shorter. Benign neoplasm usually have normal telomerase activity. Malignant neoplasm, on the other hand, have upregulation of telomerase activity. They do not lose genetic material after multiple cell divisions, and that keeps them alive. Another concept to understand is the concept of monoclonality. Benign and malignant neoplasms are derived from a single precursor cell, or described as a monoclonal proliferation. So the concept is one cell has gained the mutation to proliferate and form a neoplasm. By contrast, non-neoplastic proliferations are derived from multiple cells. They are polyclonal. So we can use this concept to distinguish whether swelling is a neoplastic swelling or a non-neoplastic swelling. So we have seen the features that are characteristic of benign and malignant neoplasm, which are differentiation and anaplasia, nuclear features, rate of growth, local invasion, metastasis, telomerase activity, and monoclonality.